Hello everyone, welcome to my presentation. My name is Marion Wels-Bennick and today I present on our model to predict the fate of Listeria monocytogenes in cheese. This work was performed together with Ellen Wemmerhoven and Marcel Zwietering. I work at NIZO, which is an independent contract research company based in the Netherlands. Together with my colleagues, we provide hands-on solutions to food manufacturers to ensure food safety and quality in the entire food chain, from ingredients to finished products. We have expert knowledge in this field and on product formulation and processing conditions. We apply our know-how to address our customers' needs. First, a short introduction on Listeria monocytogenes. As most of you will know, this pathogen can cause the serious illness listeriosis. This disease has a mortality rate of up to 30%. Young, old, pregnant, and immunocompromised people are at risk to develop the illness, which can occur upon ingestion of contaminated foods with Listeria. Clustering of individual cases and linking these with particular foods has not always been straightforward, because the time between ingestion and the onset of disease can be variable and long. But this process has improved greatly over the past decade using whole genome sequencing. Inactivation and control of Listeria monocytogenes is important to ensure food safety and prevent foodborne outbreaks. The bacterium is widespread in the environment and it can be transmitted to foods or food production environments. Listeria is a rather persistent bacterium. It can grow at very low temperatures, in high salt concentrations, and at low pH. It also has the ability to attach to surfaces and produce biofilms. This is an unfavorable trait in processing environments. Listeria is inactivated by pasteurization. Post-processing contamination must be prevented by applying good manufacturing practices and adequate sampling schemes. This holds particularly true for ready-to-eat foods like cheese. Such products do not undergo a heat treatment before consumption. In case Listeria is present, this may pose a health risk. In Europe, specific microbiological criteria for Listeria in RTE foods have been set in EC Regulation 2073-2005 for 3 different categories. Foods intended for infants or medical purposes fall in category 1.1. Other RTE foods fall in category 1.2 if the product is able to support growth, or in category 1.3 if it cannot. For products in category 1.2, Listeria must be absent in 25 grams when it leaves the production facilities, and levels must be below 100 CFU per gram during shelf life until the moment of consumption. This maximum level also applies to 1.3 products that are in the market. It's therefore important to determine whether a product can support growth of Listeria or not. For some products, it's easy to establish if growth can occur or not, based on growth limits of the pathogen. For instance, products with a pH below 4.4 or water activity below 0.92 will not support growth. If product characteristics do not meet specific boundaries for growth as listed in the EC regulation, it's possible to demonstrate growth or no growth potential via predictive modeling or by durability or challenge tests. In the US, a zero tolerance applies to Listeria monocytogenes in all ready-to-eat foods, which equates to an absence in 25 grams. While these zero tolerance levels are in place, it is still important to know which types of products may pose the highest risks. In 2003, the Food and Drug Administration published the Quantitative Assessment of Relative Risks to Public Health from Listeria monocytogenes for selected ready-to-eat foods. This includes cheeses. In the assessment, different cheeses were grouped and classified based on moisture content. Cluster analysis of predicted per serving and per annum relative ranking showed that consumption of soft unripened cheeses is of high risk. This includes, for instance, cottage cheese or ricotta. A moderate risk was associated with soft ripened cheese, semi-soft cheese, and fresh soft cheeses. Cheeses in these groups include, for instance, brie, feta, mozzarella, and blue. A relatively low risk was established for hard cheeses such as cheddar and parmesan. Gouda cheese was not explicitly listed, but would belong to the semi-soft cheeses in the FDA risk assessment, given its typical moisture content of 42%. When looking at the EC regulation, Gouda does not automatically fall in category 1.3, as it has a pH above 5.0 and a water activity above 0.94. Yet, Gouda cheese has never been associated with listeriosis, and the challenge test from 1988 showed no outgrowth. More recently, we confirmed no growth in challenge tests, and we established that the main factor contributing to full growth inhibition of Listeria monocytogenes in Gouda was undissociated lactic acid. We came to this conclusion by calculating the concentrations of undissociated lactic acid in the water phase of Gouda cheese and comparing these with the highest levels needed for full inhibition of growth in a broad range of Listeria strains. This level was 6.35 millimolar for the undissociated form. It is important to stress that the protonated form of lactic acid has the antimicrobial activity and not the conjugate base. The formula to calculate the concentration of undissociated lactic acid in the water phase of cheese can be found in our food control publication. The concentrations of undissociated lactic acid in Gouda clearly exceeded the level of 6.35 millimolar, and therefore this product does not support growth of Listeria. Other factors in Gouda, such as moisture, fat, and salt content, did not significantly contribute to growth inhibition. Based on this knowledge, we wanted to establish if undissociated lactic acid concentrations in cheeses are more generically a good predictor for growth, no growth of Listeria. To start out, we obtained published physicochemical data for 10 different cheeses and calculated the concentrations of the lactic acid in the different cheeses. We then developed a model to predict the growth in the cheeses. In addition, we calculated growth rates in the different cheese types based on published challenge tests. The predicted and the calculated growth rates at a reference temperature of 10 degrees centigrade were then compared. Lastly, we checked if the different cheese types were involved in listeriosis outbreaks. In the next slides, I will share the outcomes of this work. The average concentrations of undissociated lactic acid were calculated in blue cheese, camembert, cheddar, cottage, Emmental, feta, Gouda, high moisture mozzarella, queso fresco, and ricotta. For Gouda cheese, we already knew that the concentration exceeds 6.35 millimolar. This was also the case for feta and cheddar cheese. In other words, no growth is expected in these cheeses. We then calculated the predicted growth rates of Listeria in these 10 cheeses based on the gamma model. Gamma factors for temperature, pH, water activity, and undissociated lactic acid were included. As input values for growth under optimal conditions, we used growth in milk at 30 or 37 degrees centigrade. The average and the maximum values were extracted for this in Combase. Input values to calculate the different gamma factors were retrieved from public databases. In addition to growth rate predictions, we calculated growth rates of Listeria in the different cheese types based on published challenge tests at a reference temperature of 10 degrees centigrade. This slide shows the predicted and the experimental growth rates at a reference temperature of 10 degrees centigrade in different cheeses. Clearly, this figure requires some explanation. 6 soft cheeses are listed from top to bottom. Per cheese, the predicted growth rates are indicated with arrows based on the gamma model calculations. Black arrows show the calculated maximum growth rates at the reference temperature of 10 degrees centigrade, and the gray arrows show the calculated average growth rates at 10 degrees. In addition, the growth rates of Listeria in or on the cheeses at 10 degrees are presented with squares or triangles. So these rates were calculated based on the actual growth rates and challenge tests upon artificial contamination of the surface, as shown with squares, or in the milk or the curd, as indicated with triangles. This shows that for all 6 cheeses, growth rates were above 0 under experimental conditions. I realize that this figure may be a bit overwhelming, but the take-home message is that the model correctly predicts outgrowth as observed in practice in all of the different 6 soft cheeses. Now, if we look at the same representation for feta, cheddar, gouda, and emmental, we see that no growth is predicted for the first 3 cheeses by looking at the arrows. Here, we predicted a growth rate of 0. The challenge data also did not show growth in feta and gouda. For cheddar, no growth was reported in most studies. 2 outlying data points were indicating growth as shown in red, but the underlying study provided no information on the pH, the acid and the salt content in the cheddar. In the case of emmental, no growth is seen in practice. It is known that lactic acid is converted to propionic and acetic acid in such cheeses, which will subsequently have anti-Listeria activity. Overall, we can conclude that a model based on the gamma factors pH, water activity, lactic acid and temperature predicts well for growth of Listeria in cheeses that contain lactic acid as the main acid. The model could be extended for cheeses with other antimicrobial factors, such as acetic and propionic acid. We also made an overview of reported outbreaks of listeriosis that have been associated with cheese. Between 1983 and 2019, at least 41 outbreaks have been reported. These were primarily due to consumption of soft cheeses and mold-ripened soft cheeses. Our study once again confirmed that these cheeses are able to support growth of Listeria. For Listeria, it is clear that chain control is pivotal and that CCPs on inactivation and prevention of post-processing contamination and outgrowth in the product in the chain are critical. Predictive models are powerful tools to assess potential risks associated with pathogens in the food chain. The current model helps in the assessment of growth of Listeria in cheeses that contain lactic acid as the main acid. Such knowledge is also applicable to other products. Over the past years, we've seen a strong increase in plant-based foods, and NISO is actively working in this field. We support food companies with development of plant-based dairy alternatives and also provide our safety and quality expertise. Lastly, I would like to thank Ellen Wemmenhoven, who now works at Arla in Denmark, and Marcel Zwietering at Wageningen University, and the Dutch Dairy Organization for funding the work. The presented work has just been submitted for publication and is part of Ellen's thesis. If you are interested in a copy, please email me. I hereby would like to thank you very much for your attention, and if you have any further questions, please feel free to get in touch.